very much for the invitation. It's a great pleasure to join you here. I feel honored to have been invited to participate in this colloquium, since I usually wouldn't qualify myself as a mathematical theoretical biologist. I'm an algebraic topologist originally, well, still, and over the last 15 years or so have become more and more involved in doing various applications of algebraic topology in many areas, particularly life sciences and especially neuroscience, but also to a certain extent in cell biology and cancer biology and so on. And that's the work I'm going to be talking to you about today, relatively recent work, which is done in collaboration with Kelly Max, who's a former PhD student of mine, Marcus Youssef, who's still a PhD student of mine, a couple of PhD students in life sciences at EPFL, Cyril Pulver and Jovan Isma, and Fan Nguyen. Mutab Harthaus, who's one of my professor colleagues at EPFL, and Gianpaolo Dotto, who is a professor at the University of Lausanne. And so Kelly and Marcus were both financed by an EU ITN grant, Cancer Prize, for most of the research on this project. Okay, so I'd like to talk to you about the kinds of challenges that we want to address with respect to single cell data and how topology and geometry, which are two kinds of mathematics one doesn't often think about in the context of cell biology, actually can play an important role in analyzing this very complex and noisy single cell data. So one of the challenges we face is being able to identify cyclic processes. So these can be processes related to returning to homeostasis. It can be the response of genes to cycling hormones, for example. And we'd like to be able to pick out these sort of cyclic processes in single cell data. So the method that we apply is a method of gene enrichment, so enriching these data sets via topology. So topology is the mathematics of shape, right? And so it seems that maybe if you use that kind of mathematics, you can be able to pick out these kinds of geometric structures. So more precisely, use a tool from applied topology is called persistent cohomology that I will explain. And so the tool that we build is a pipeline that enables us to detect and then compute the statistical significance of circular structure in single cell data. So if we do a little bit of a cartoon, we can imagine that we do PCA projections of the gene expression data for different gene sets onto the principal PCA components, and we look for cyclic structure in those. So I take my cursor here. So here there's oh yeah, that looks like some sort of obvious circular structure here. And so we have the maybe different expression profiles or something like that that are giving us the colors of the cells. Or you could take another gene set and you say, there doesn't seem to be much of a circular structure there, or even less here in the projection, PCA projection related to this other gene set. And then using the tool of persistent cohomology, one comes up with a sort of topological signature for this data. We call it persistence diagram. And I'll explain something about where these diagrams come from. But the point is that you can find it could be a sort of a signal of interesting circular structure that one can find in these persistence diagrams, here highlighted with one of these red circles. And you can say, okay, there's a hint that there's interesting circular structure there. Can we measure its significance? And then I'll explain to you a statistical test one can use to determine whether or not the signal that one reads from the persistence diagram actually is picking up some meaningful circular structure. And so we can measure the statistical significance. The other challenge that we deal with is once you determine one of these circular processes, how can you find what cascade of genes, so one gene expression leading to expression another gene, leading to expression another gene, that underlies these cyclic processes? So here, the method that we use is actually going to use geometry now. So we're going to look at how the gene expression structure is really embedded in the whole phase space of the transcriptome, the geometry of that embedding, and from that be able to infer something about the sort of cyclic or pseudo-dynamics, which is sort of inferring some sort of time component, some sort of temporal component. So from the geometry of this embedding, and that's what I'll explain. What that gives us then is a pipeline to estimate, once you have this circular structure, really what the phases of the different cells and genes are as you evolve through the circular structure. So the idea here, I'll come back here again, is sort of a cartoon. We're going to end up with some sort of matrix, a nice skew-symmetric matrix, from which we're going to be able to read things about what are which genes are leading which other genes. We'll look at projections on various planes that will pick out sort of the dynamics of the relations between the expression of two different genes. And from that, be able to describe the different phases. So as you go around the circle, you can look at in what phase of the circle you have the different genes that are active, and something about the amplitude of the expression of those genes. And so we'll be able to say is if here we have time along the x-axis and gene expression along the y-axis, you're looking at three different genes here, you could say, well, we see that there's a bump in gene expression of gene I, which leads that in gene J, which leads that in gene K. And this we're also seeing in this circular representation here. So that's what we're going to be able to pick out from the geometry of this embedding. So there's an underlying philosophy here that I think is important. So here we have this Waddington epigenetic landscape about the evolution of gene expression and how it can sort of lead you into valleys from which you can't escape and so on. And the point is that there's a very complex geometry to this epigenetic landscape. And our philosophy is that the topology and the geometry of this landscape actually have biological meaning. And I hope to convince you that we have some strong evidence for this. So if we think about paths in this landscape, they correspond to developmental trajectories. So as, for example, you have a fetus developing, or you have a cancer developing, or something like this, you have a developmental trajectory in gene expression in this epigenetic landscape. But then you could have loops in this landscape. You could have processes that come back to themselves. And so loops in this epigenetic landscape correspond to cyclic processes. And the nice thing about using topology is that topology is more robust. It's less sensitive to scale, to exact placement, to translation, and so on. And the stability that comes from using topological features of the data enables us to ensure that we have detected robustly various cyclic processes. And when we think about single cell data, I think a good way to think about it in this context is as taking a finite sample of this epigenetic landscape. And so that's the underlying philosophy of what I'm going to be discussing. So now I'm going to go back to these challenges we faced, the first of which was the identification of these cyclic structures. That's what we call circular enrichment of the gene set. So I'm going to give you first an overline of the pipeline that we built, and then go back and explain each step in more detail. So if we're given a single cell data set, the first thing we want to do, there are about roughly 20,000 genes in play. That's really too much for to analyze at any one time. The idea is to focus on particular hallmark gene sets. So these are ones that the cell biologists know about, where they've said, okay, these particular genes we know or we suspect strongly are related to these kinds of processes. And so maybe you have 50 or 100 genes instead of 20,000. And when you restrict this hallmark gene set, then you have obviously a much smaller, your point cloud is embedded in a much smaller space. And so it enables you to detect, the circular structures you're looking at for more easily. And it also, because these hallmark gene sets are annotated, it can give you a preliminary interpretation of any results that you might get. Now, that's still too many dimensions in which to work in order to be able to do any reasonable computation. So we're also going to then project onto the first k PCA coordinates for some appropriate k, which usually take to be 5, because that's something that's easy to deal with and has already given us nice results. There's no real mathematical justification for choosing k equals 5. Then what we do, and this is what I'm going to explain, we're going to compute what's called one-dimensional persistent cohomology. So we've taken this very high-dimensional gene expression space, so this is a point cloud in 20,000 dimensional space, restricted first to this hallmark gene set, so maybe down to 50 or 100 dimensions, and then project onto 5 dimensions. So we now have a point cloud embedded in 5 dimensions, a collection of points in R5, and we're going to apply these tools of persistent cohomology to look for relevant circular structures. Then the next step, we want to see whether the circular structures that we detect are actually significant. To do so, there's something called a ring score that we can compute for this one-dimensional persistent cohomology, which we can then use to establish the statistical significance or not of the circular structures detected. So that's what I want to explain now. I'm going to explain, starting at step 3, because the first 2 are really biology. So here's the idea. We start with some point cloud here. So it's some collection of points. Here we're just in R2. And in order to compute what's called persistent cohomology, what one does is start with little balls that are centered around each of the points and slowly grow them and see what sort of geometric structure comes out of that. Because you just have a discrete set of points, there's not much you can say using the tools of algebraic topology or geometry about this. So to integrate this more sophisticated mathematics into just a collection of finite points, what one does is consider a sequence of geometric objects that one obtains by starting with little balls centered on each point of the same radius and slowly having the radius grow. We'll come back to another, I'll illustrate this process again in a moment, but here's the basic idea for 3 different possible radii. And so looking at this picture, you can say, well, there seem to be, if we just look at the black dots, you can say, yeah, there are probably 2 interesting circular structures there. They're more or less clear once you've grown the little balls to this radius. If then the radius is quite a bit bigger, then you say, oh yeah, here we have these sort of 2 annulus structures here, so 2 circular structures. And then when the radii have grown even more, then the hole in the middle is filled in and the 2 blobs have belonged together. So one thing, when looking at a picture like this, there are 2 things you could look at. You could look, for example, at how many different connected components you have. And that's what's being counted here with these sort of red dots, which correspond to also looking at the dendrogram, is just like how many connected components do you have and at what radius do they all join together. What we're more interested in here, because we actually want to compute, we're looking for circular structure. We're looking at what are called homology classes of dimension 1, or otherwise these sort of circular structures. And what the points in this persistence diagram here correspond to, on the x-axis, what's called time of birth, is the radius at which, the radius of the little balls, at which you first see these complete circular structures appear. And on the y-axis, you have the time of death, which is the radius of the balls at which they've just filled in this gap here and the circle disappears. So here we would have these 2 points here correspond to the fact that you have these 2 circles that appear at some radius when all the little balls are filled together. And then at some point, the hole in the middle is filled up and those classes disappear. And that's what these are. And then what one does, one can translate this into looking at the total persistence of these classes. So it's the time between birth and death. And so what we will call the ice persistence is the difference between the time of the death of the ice class and its birth. And here the order is given by the total persistence, so P0 greater than equal to P1 and so on. So this would be P0, P1, P2, and so on. So this is just a sequence of numbers that one can extract from a point cloud like this that are actually very revelatory of the geometric structure and potential circular structure of such point clouds. So let's look at this again with another picture. We'll go more slowly through the growth of these radii. So here the black points are my point cloud, and I want to understand its structure by putting these little balls around the points and having the radii grow. All the radii grow in parallel, and we'll see what happens to this geometric structure. And what we do is, when 2 of the balls belong together, we're going to join the 2 nodes by an edge. And what we're going to focus on is the collection of points and edges that we get in this way. So this is with a radius of 0.3, and I call this VR because it's called a Vittorius-Rips complex. So this is a Vittorius-Rips complex we get for this point cloud at radius 0.3. If we increase the radius to 0.5, then we see a small circular structure appear. But if we go back to 0.3, we say, but actually there's got to be this looks like there's a hint of a bigger circular structure. So maybe this small one that I get here is interesting, but maybe it's not the most significant circular feature I have. I increase again to 0.7, and I say, okay, now I really see there's this bigger circular structure that's really starting to appear here. And recall that every time I have an intersection of 2 balls, I put an edge between the nodes. We increase to 0.9, and there it is. There's this big circular structure that we sort of hinted at when we could just see the points themselves. And so this is what we would call the critical threshold radius. It's the radius at which this significant circular structure is born when it first shows up. And we'll keep that one in mind. 0.9 was our critical threshold radius. And we continue on, 1.1. Okay, we still see the hole in the middle. 1.5, we still see the hole in the middle, but we're starting to have quite a lot of edges, nodes that are joined by edges. And then, oops, we get to radius 3, and all of a sudden it's all glommed together. Every single pair of points is connected. So let's go back now to this critical radius of 0.9. So this is where this structure first appeared. So the way that one encodes this kind of circular structure is by what's called a one cocycle. So a one cocycle is an element, we'll call it alpha, of the real vector space that I'll denote like this. So it's a set of functions from the set of edges, so all the edges of this structure here, to the real numbers. That is, I'm assigning a real number to every edge in this simplicial complex, actually, in this graph. We assign a real number to every edge in the graph that we've constructed here. And because it's a circular structure, it'll turn out this alpha here satisfies a nice property. So it's not just what we call a one cocycle, but one cochain, but actually a one cocycle. One point I want to make here, because we'll come back to something about this, is there are actually infinitely many equivalent ways to choose this encoding as a one cocycle. So this vector space here, it's a vector space of functions to the real numbers, where the vector space structure is just inherited from the addition and multiplication in R. It's called the vector space of one cochains on this graph VRR. And alpha is a one cocycle. So it's an element of this vector space that has a nice property. So it's what we call one cocycle. And there are infinitely many ways we could be choosing this. We choose one. And then we'll figure out how we can choose the best one in some sense. Okay, so now that we've done this, we need to say, is this structure actually statistically significant? And the way we do that is by computing what's called the ring score. So here, we're associating to this, we had this set of persistences. We had P0 greater than or equal to P1 greater than or equal to, and so on. Let me just go back to that for a second. Here, right, so we had P0 greater than or equal to P1 and so on. So this, I'm going to look at this sequence that we got from our persistence diagram, and we're going to plug that into a formula for what's called the ring score. And what we do is we look at the difference between P0 and the sum of all the other PIs. Now, there won't be an infinite number. There'll be some arbitrary, some large finite number, perhaps. And we subtract this sort of sum here. The point is this first persistence should be the most important, and we weight them so that we take less and less account of the other ones. And we're going to get a number that's always somewhere between 0 and 1. The greater this number is, the more significant the circular feature. If we applied it to something that was really a circle, we would get 1. And the closer it is to 1, the closer it is to be something that's actually circular. And importantly, because we're thinking about things logically, this ring score is actually scale invariant. If I take the same point cloud and I blow it up or I shrink it down, I'm going to get the same ring score. So again, the closer the ring score is to 1, the closer it is to actually being something that is a real circle. So how do we use the ring score to determine the statistical significance of what we have? So suppose we have a ring score for a point cloud of expression data. So I've chosen some N genes. I look at their gene expression, and I have the point cloud of expression data. So I have a point cloud, a collection of points, finite set in Rn. And then I do this persistent cohomology computation. I found a circular feature that looks significant. And I say, okay, how am I going to test its significance? And what I'm going to do is a permutation test that's gene set based, but preserving the correlation structure of the gene expression data. So what we've done is to randomly generate 2 to the 10th sets of N genes and compute the associated ring score. And so that's going to give us a null distribution of the ring scores. And then with respect to that, we can compute an empirical p-value for the original ring score to see whether it actually could be significant. So that's how we detect and then measure the statistical significance of circular structure in point clouds in general, but in point clouds of expression data in particular. But once we're given the circular structure, we'd like to now say something, extract some biological information about it and be able to see, okay, what exactly is the cyclic process? Which genes are involved? In which order? According to which amplitudes? So the pipeline we put into place is like this. So suppose we detected a statistically significant circular feature, which you've encoded by one of these one cocycles. So it's a function from the edges of VRR. to the real numbers. And what we want to do is to then assign phases according to this circle to the individual cells. But we need to find a way to do that sort of in a unique manner, a manner that makes geometric sense. And that'll be by the choice of what we call a harmonic circular coordinate. And I'll explain how we go about finding that. Then once we have the circular coordinate, we want to figure out what the different phases or offsets of the gene expressions are and the amplitudes of the gene expression. The way we do that is by something called a lead lag matrix, which looks at the pairwise gene ordering of the different genes with respect to a chosen harmonic representative of the code cycle. This is getting a little more technical. We'll go through it slowly. And I'm not going to give all the details, but we'll see something about how this works. That's our goal. So what is this harmonic representative story? So suppose, back at our critical threshold at which we first see significant circular structure when we're looking at this series of topological spaces or geometric objects that we get by slowly growing these balls around the point. So this is in the case we were talking about before. This is R equals 0.9. Now, we've already talked about this vector space here of functions from the set of edges to R. So that's the set of the vector space of one cochain. We can also look at the functions that assign real values to the nodes of this graph. We'll call them zero cochains. We look at these two real vector spaces here, and we can choose inner products. They're real vector spaces. We can define inner products on them, and we define inner products that are chosen to correct for the fact that the point clouds that we have when we're looking at gene expression data are actually highly non-uniform in terms of their density. And there are ways of fixing an inner product so that you can kind of correct for that density. And you do so on these real vector spaces. Okay. Once we have inner products, we can do geometry. Inner products give us notions of angles. They give us notions of norms. So we have notions of distance that enable us really to think about things in terms of geometry and not just purely in terms of shapes without any notion of distance or angle, which is topology. Once we have fixed those inner products, then we can compute what's called a harmonic representative of the circular structure. So we started with, we know we have some significant circular structure. We know that it can be encoded by an element of this vector space here, which will be a one cochain. And there's a very special one called the harmonic representative, which is chosen with respect to our inner products here. It's a unique choice. So where there's, the unique one that is harmonic. There's no more choice involved, which is something we always like as mathematicians. And then that'll be our representative of the circular structure. And with respect to that, we can also define a circular coordinate that is going to be going to give us a map from a function from this VRR, so this collection of points and edges, to the circle S1. And we choose it so that it has the smoothest gradient with respect to these chosen inner products. And there's a close relationship between this alpha H and this theta H. But the nice thing is that we have, in each case, there really is a specific choice that we make. There's not like we choose one at random. And that helps us, that ensures that we're really staying close to the geometry, and I think probably enables us to have more biological relevance. And then what we can do once we have that harmonic representative is to determine the phases of the points in the point cloud, so the phases of the different cells using this theta H. So how does that work? So we're back to our example here where the critical radius was 0.9, and this theta H is giving us some way of defining a function from this VR 0.9 to the circle S1. Okay, so on this side we have our VR 0.9, and this side we have the circle S1. And we have here some function from here to here. And how does this give us phases? Well, we can think of the different points in the circle as giving us different angles or different phases. And we can see, okay, what is the pre-image of those points? So if we start with this blue point here on the circle, then we see, okay, well, that corresponds to point here. It gives me the phase of that particular node. If I look at the green, then it gives me the phase of this node here. And if I look there, then there are actually two nodes that are in the same phase there. And so you can do that sort of all along and determine sort of associate a phase to each of the points in your point cloud in this way. So if you go back to a cartoon like this, then we have this sort of complementarity between the circular coordinate and this harmonic representative of the circular structure. So here we can imagine we have our point cloud in 3 space. And the circular coordinates, now all the cells here are colored by their phase along the circle. And that's the sort of thing that we get from the circular coordinates. On the other hand, this alpha h, which is assigning a real number to each of the edges, we can think of as giving us a vector field. And so it's telling us something about the dynamics, because by assigning a real number to each of the edges, if I go back to this picture, for example, if we can imagine assigning a real number to each of the edges, you can think of that as giving us a vector field. It's telling us something about the gradient going from this point to this point if we're assigning a real number to this edge. So that's what this alpha h is doing here. They're providing us with complementary information about the possible dynamics. So that's already giving us some very nice qualitative information about the gene cycle, about which genes are sort of where in the process and so on. But we'd like to sort of nail it down a bit more, become even more quantitative, and that's where we really get more serious about the geometry. So the geometry is already coming in here because we're using these inner products that are on the basis of 0 cochains and 1 cochains, which are really coming, linked to this, the question of the density of the point cloud. But we really want to get even more serious about the geometry. And that's where we start with what's called lead lag analysis. So let's think about a very basic picture here, where we have 2 different curves. This is the yellow curve and the blue curve. They're basically the same shape, but one is shifted with respect to the other. So if you look at these 2 curves, you could then translate what you see in this picture on the left to the picture on the right, where now you're plotting blue against yellow. And when you plot blue against yellow, it's giving you a curve now in the plane R2, right? So this blue curve here, which maybe should become, is just comes from plotting for any value, the blue against the yellow. And when you do that, so it's giving you some function gamma from the circle into, in this case, R2, but more generally, it could be into Rn. If you had some sequence of n functions now, and you did the same sort of thing, it would give you a map from S1 into Rn. And often, you can actually extend this function from S1 over Rn to a function over the entire disk, the disk of which the boundary is the circle. So this is the sort of thing we're going to analyze, which has already been this kind of thing just for paths in the plane or in Rn has been analyzed by Baryshnikov and collaborators. They provided the first tools for doing this sort of thing. The idea is you look at a vector field or one form in the plane like this. So this is sort of the angular velocity vector field. And you can integrate along this curve with respect to this vector field, which by Stokes' theorem is the same thing as looking at the area, signed area inside this curve. And the signed area of the region of the plane that's bounded by this curve, so this curve is determined, has its components gamma I, gamma j. So we're starting with our function gamma from S1 into Rn. We fix the coordinates gamma I, gamma j. And we look at the curve in the plane xi, xj that's determined by the components gamma I, gamma j. And the signed area determines the extent to which the function gamma I is leading the function gamma j, is ahead of the function gamma j. If that area is positive, gamma I is leading gamma j. If it's negative, gamma j is leading gamma I. So this is the lead lag idea. So again, if we're starting, there's a particular example where this is easier to analyze with what's called the chain of offsets model for when you have a situation where your gamma, which is going from S1 into Rn, satisfies the following. So you suppose you have some vector A of real numbers. You have another vector B of numbers between 0 and 1, strictly less than 1. And you have a cyclic function, chi, from the circle into the real numbers. So that when you evaluate gamma j for any component j here on Z, which is an element of S1, you can think of S1 as a subset of the complex plane, then there's this very simple expression for it. So it's some shift of the function chi by beta g, and then also the amplitude modified by this factor aj. So you have all of the gammas that are sort of different multiples and shifts of the same cyclic function. And that's what you call a chain of offsets in this case. And so you'd like to know, with respect to this chain of offsets, is gamma I leading gamma j, and so on. This is a very clear case of what you're looking for. In the case when chi is very simple, you're just multiplying by some complex number, then it turns out that this Lij that we talked about, which is this lead lag coefficient, is actually proportional to the sign of 2 pi bi minus bj. So it picks up this shift, this offset between the ith coordinate and the jth coordinate. When you do this, you get a skew symmetric matrix that's encoding these offsets between the different coordinates. And under reasonable conditions, it picks up things about the amplitudes as well. So what we want to do is generalize this sort of lead lag analysis to thinking about the function we have from VR to S1, because that'll tell us something about, we'd like, because remembering that the nodes of this VR graph correspond to the different cells, and we want to know something explicit about their phases and about which genes are active. It makes sense to try to generalize this to thinking about the complex we were studying. So here's a reminder of the picture we use when we're thinking about leading and lagging, which coordinate is leading which coordinate. So now suppose that we have our harmonic representative of our cyclic structure. So it's some function from the edges of VR through the real numbers. And what we're going to do is choose some affine embedding, so it's a nice geometric representation. We're going to take this VR and sort of embed it into Rn for a large enough n so that it's really all of the points are embedded in an affine way. You can always find an n big enough to do that. And then for any pair of numbers between 1 and n, we can define a function that has this funny name, so an integral blah, 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 from the set of edges of VR to R. So in other words, it's going to be a one cochain. And it's defined in the following way. It's going to take an edge of this complex to the integral of this angular velocity differential form, which we already saw, xi dj minus xj di, along the path that we get in Rn is determined just by the embedding of the edge. So if I look at an edge in my diagram, I've embedded it into Rn, it's going to give me a path in Rn. And I'm going to integrate this differential form along this path. And that's going to give me a real number. And so what I've done is assigned a real number to every edge of VRR. And that's exactly what a one cochain is. Now remember that I have chosen an inner product on the vector space of all one cochains. And I'm going to use now that inner product in order to think about leading and lagging. All right. So we have our harmonic representative that we have our affine embedding that we looked at. And now we're going to define a lead lag matrix with respect to this embedding, with respect to this choice of our harmonic representative. And the coefficient is going to give me an n by n matrix. And its coefficients are defined as follows. The ij coefficient, with respect to the embedding I chose, with respect to this harmonic representative that I chose, is given by the inner product between AH, which is a one cochain, and this one cochain that I just defined, this one where I'm doing integration along the path determined by an edge in Rn. And since I have these two one cochains, I have chosen a while ago an inner product on the space of one cochains, and I just take that inner product. That's going to produce a real number. This matrix Lij is skew symmetric. Not hard to see that it's skew symmetric, and therefore one can do a spectral analysis. You can look at what are the complex eigenvectors and so on. And by doing sort of generalizing the lead lag analysis that was developed by Baryshnikov et al., you can see that you can use by looking at the complex eigenvectors and the largest principal eigenvalues of this matrix, you can infer from that the ordering and the amplitude of the activation of the genes. That gets a little bit more technical, but we'll stop there for now. The basic idea of how we can do this. We're generalizing this lead lag analysis along just one curve, which is for just two coordinates, which is something that is quite well understood and enables us to look at the pairwise ordering of the different genes and their expression. So here's the idea, that we get this lead lag matrix by looking at all these pairs of genes and seeing which one is leading which one as we go along. It gives us this huge symmetric lead lag matrix like this, using this integration along the edges of the angular velocity differential forms. And then once we have this lead lag matrix, there are two things we can do. We can project onto the principal complex lead lag planes like this, and that gives us the planes where there's sort of the most circular action going on. And then we look at the components, and that puts out which genes are active and when, according to the different phases. So it really provides a more quantitative picture of the gene phases and their amplitude. And so it's going to produce in the end these sorts of curves where we see, this is where gene 1 is active, gene J, gene K, and so on. All right, so that's the theory. Let's see whether it really works. Let's do some validation. So we look to apply this first with synthetic data. So we're using a linear ODE model of gene spread, transcription, and translation, taking into account the fact that RNA transcription apparently has some bursty behavior. So here's the model where we're looking at the vectors of mRNA and protein level. And we have an interaction matrix describing the effect of protein J on the RNA production of gene I. And then we have matrices corresponding to degradation rates of RNA, translation, and degradation rates of proteins as well. And the bursting behavior we take into account with the threshold function sigma. And then in order to ensure the existence of circular structure, we're going to make a specific choice of A, which is precisely builds in this sort of cyclic structure in the by giving us a rank 2 matrix. Okay, so that's the model we're going to look at. We want to see whether we can detect properly the cyclic structure, the phases, and so on. So, and then because we want to have something that looks more like actual noisy single cell data, there's something called negative binomial sampling, which was came out in Nature Methods last year, which we use in order to make the data that we analyze look more like actual noisy single cell data. So first, we start looking at the results. We see here's on the left, the interaction matrix, which is really, the actual, the ground truth. And then what we estimate from looking at the lead lag matrix, you can see that the estimate actually matches very nicely with the underlying the real data. We could say, okay, that's good, but how about detection of circular structure? So by looking at the circular enrichment, we see that we had quite nice ring scores when we're looking at PCA components 1, 2, 3 and 1, 2, 4. This, for example, is showing us the projection onto PC1 and PC2, that there's this very nice ring structure that we see here. The color here is corresponding to the coordinates or the phase as we go around the circle. So you see that there's a very nice presentation of this. And then if we look at, we can do one of these charts to give a phase the different genes and see how that matches up with the circular structure that we had. And you can see that it matches. There's a nice match with the circular structure as we go around and with the expression of the gene. And we like to see, okay, well, we know actually, because we have the ground truth, we know what the actual phases and the actual amplitudes are. How well do our estimates match up to that? And so here we're mapping the actual index of the gene, which is giving the phase, against the phase that we detected. And we see a very, very good correlation, 0.986. And then the amplitude's not quite as good, but not too bad. But the phases work extremely well. We also did a validation on actual cell cycle data. So we took data from cycling human dermal fibroblasts. And the hallmark data sets that we used were known markers for the S and G2M phases of the cell cycle. And we got a point cloud that looks like this when you look at the lead lag. And you see the S part of the cycle that's very active here, and the G2M part of the cell cycle that's very active here. So it seems to be nicely spread out like this. We again can look at the lead lag matrix, and we see a nice, the S leading G2M as is expected. plots very nicely in the phase plot as well, with S followed by D2M, so that matches well with what one knows about the cell cycle. And then you could look for specific genes that are MCMC, HELS, and so on. These are various genes for which you say, okay, when are they most expressed? And you can see when exactly they're expressed in the different phases of the cycle like this. And you can also look at, here we're plotting the gene phase, or here along the x-axis against gene expression for a bunch of different genes, and you can sort of see how the activity of the genes is shifting from left to right. So briefly, one actual experiment that we did once we had the validation was to look at prostate regeneration. It's androgen induced. So here we wanted to actually test this for in vivo data and multiple RNA-seq samples. This was coming from a paper of Carthouse et al. In Science 2020. They had single cell data from prosthetic epithelial cell regeneration. And so the idea here is that the castrate mice on day 0, on day 28, the prostate would have shrunk quite a bit, and you add testosterone back, and the prostate regenerates. We tested this against more than 50 hallmark data sets. And for 2 of them, for example, here is the androgen response and cholesterol homeostasis gene sets. This is these persistent pictures here. And we have persistent and significant cellular structure that we see in these persistence diagrams, highlighted in red here. And indeed, they have the p-values of the ring scores are very low. And so we really do have significant circular structure that's coming up here, which is not surprising. We have an actual cyclic process. But this is detected with respect to multiple hallmark data sets, not just these 3. This is just an illustration of what we got. And then you could look again, you can do this sort of plot where you see you're going around the circle, you're seeing the phase of the different cells in terms of their gene expression, in terms of the level of gene expression and so. Again, this is all with respect to the androgen response gene set. And again, using the lead lag projection, you can see very clearly the phase as you go around with respect to the various coordinates. And then here I'm matching it against the colors here correspond to cells that are taken at different time points of the regeneration process. And you can see that there's a decent correspondence between these and the different phases that we get from the mathematics. And just to compare, you can also do the same sort of thing for the cholesterol homeostasis gene set and get the same size of nice matching. Finally, you can look at the same kind of phase versus gene expression for various genes, and you see again this sort of shift, very nice shift of the gene expression. This is all coming from this lead lag analysis. So to conclude, topology and geometry enable us to identify significant circular structure and to infer gene cascades underlying the circular structure. Advantages to this approach: no ML technique, no parameter fitting. It's applicable to a wide range of experimental data, heterogeneous samples, multiple data sets, and the topological signals are in fact robust across data sets. There are limitations because there are a bunch of hyperparameter choices in the pre-processing stage, as always. And there's a potential impact of outliers because the choice of persistent cohomology tends to be sensitive to outliers, but there are ways to deal with that. Future directions that are under consideration, we'd like to integrate RNA velocity, which is an obvious thing to study in the context of single cell, into this pipeline. We'd like to think about how do you have a theory, a mathematical theory to describe concurrent biological processes. Here we were looking at one process at a time. We'd like to think about processes going on simultaneously and to analyze a lot more experimental data. And with that, I'd like to thank you very much for your attention, and we'll be happy to answer any questions.